Bueno, mañana les recuerdo que es el examen y ya pueden descargar las presentaciones inclusive de la clase de mañana para que la lean y la revisen. Porque la clase de mañana va a terminar a las 3:30 y a esa hora se les va a dar el examen, ¿sí? Y se les va a dar como unos después de las 4 se les va a dar todavía alrededor de 15 minutos para que puedan terminar, ¿de acuerdo? Bueno, pues entonces nos vemos al rato. Ok.
Okay, so hi everyone. Let's start where we stopped yesterday, and we talked a little bit about different pressures that we have in anaerobic digestion that might cause problems. For example, temperature, ammonium, conductivity, pH. So there are several stressors that we mentioned. And then we went into some studies which are quite applied, research studies where you could follow taxonomic profiles in regard to the chemical parameters. So we tried to discuss these things together. And yeah, we will continue with this today. Maybe I will shortly summarize with own words what we have seen yesterday. So we started with the question whether we could actually apply a thermophilic dark fermentation without any adaptation time, and apparently this is not possible. We thought it might be possible since bacteria grow much faster than methanogenic archaea, and usually we don't have methanogenic archaea in the dark fermentation, at least we don't want them there. So we assumed that the bacteria might adapt much faster, but this was not the case. So also in dark fermentation, we have to adapt very slowly, step by step, increasing the temperature in order to allow the microorganisms to work sufficiently. We were then asking whether it might be possible to apply transient heat shocks. And this was quite interesting because the microbiome was relatively robust against these transient heat shocks. And when we compared it to the control, it did not make a big difference whether we applied heat shocks or not on the final accumulation of organic acids and the degradation efficiency. But we saw that the methanogens might be inhibited due to the heat. So as we want to inhibit the methanogens anyway in dark fermentation, and usually we do this with pH, this is a nice opportunity to apply another selection pressure to selectively inhibit the methanogenic archaea. Well, we were then continuing with dark fermentation, and we started to talk about ammonia, about chicken manure, which is quite enriched in nitrogen. And then we saw that it's quite problematic to perform dark fermentation with residues from chicken manure because due to the ammonia, we have a stabilization of pH. It's a quite efficient buffer. So we have a pH around 7 or so, and then the methanogens can again work, although we want to make a dark fermentation. So this was not working very well. So we wanted to improve this. And we tried an alternative approach, so we increased the loading rate very much, and this was possible. This allowed us to inhibit methanogens even though the pH was not low. This was interesting, and then we also thought about the combined application of nitrogen stripping methods, because if we want to use the residues from the dark fermentation, it might be good to control the nitrogen level before it goes to the methanogens. And well, in this regard, we saw that there might be an inhibition due to the salts that we have to add for the nitrogen removal technologies, like for the MAP precipitation or the nitrogen stripping. And well, based on this, we assumed that there might be an inhibition on methanogens based on the gas production, but this was not so clear. So this was the gas production, if you remember from yesterday. And well, the difference is just slightly. So you might assume that in the case of a 3 times MAP precipitation, the amount of methane is slightly reduced compared to the control. But since we are working with very low amounts of gas and there was not a high rate of repetition, it was very difficult to make this significant. So we wanted to go for another approach, and this was based on qPCR. Anyone has ever heard about qPCR? No? 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 Okay, maybe you remember the PCR, the polymerase chain reaction. It's a possibility to multiply DNA sequences. And with a qPCR, we can make this quantitative. We can say how often a certain DNA strain was amplified in order to get an idea whether a certain gene is very abundant or not. And we are performing this qPCR. I will not go into the methodological details because this would take some time and we are already a bit late with the lecture, so I try to finish the most important parts. And I will just jump over the methodology for the qPCR, but just remember it's a methodology which we can use to quantify genes or certain DNA fragments in our samples. Well, we took probes for archaea. This was quite interesting, and you see that in the control, the abundance of archaea was still relatively high, although we applied high concentrations of nitrogen. So it suppressed the methane production already, but yeah, there were still some methanogens given. Hi, come in. So, and then we see that depending whatever technology we apply, whether it's the nitrogen stripping or the MAP precipitation or the 3 times repeated MAP precipitation, the number of copies of the gene for the archaea that was used went down. So this is another option to inhibit the methanogens now. But you see already it's not too easy to get rid of these methanogenic archaea. We were also wondering whether the bacteria are inhibited. And here you see the control. And then with a one-time NH3 stripping process, there are no changes. So bacteria are similarly relatively robust. And then with the MAP precipitation, it looks similar. Maybe here gets a bit down. But then with the 3 times repeated MAP precipitation, the bacteria are also going quite down. And I was surprised by this because if you remember this picture, we saw that for all treatments, much more acid was produced compared to the control. So although bacteria were inhibited here, they seem to produce more acids than in the control. Yeah, so this was quite interesting. We also went into taxonomy, and well, this is quite difficult to analyze. You see many taxonomic names here. We went already on the genus level. And well, since there are so many, we also numbered them in groups. And the most important thing that you see here is that the numbers on the first 3 treatments, the control, the NH3 stripping and the MAP precipitation, appear relatively regularly. But then in the 3 times repeated MAP precipitation, suddenly the numbers become a bit unordered. Everything seems to get out of order. So this could also be a hint on a taxonomical level that here was an inhibition of the involved microbiota. So what do we take home from this? So we have seen that a significant increase in the loading rate allows us to inhibit methanogenesis, even though pH does not really get down. we have seen that we can apply a further inhibition due to the MAP precipitation, especially if we repeat it 3 times. And one reason for this could be the high conductivity. So maybe you remember that we have an inhibition of methanogens already at 35 millisiemens per centimeter. And then the bacteria are more robust and they sustain up to 80 millisiemens per centimeter and can survive a higher conductivity. So we also have seen if we apply these combined methods of dark fermentation, of nitrogen removal, that we are actually removing some important trace elements, which could be important for the fermentation process. So it might be important to add them separately. And we have seen that we, by controlling the nitrogen level, can actually optimize the yield in organic acids. So in all cases where we applied the removal of nitrogen, the yield of acids went a little bit higher. We also wanted to analyze whether it is possible to evaluate based on the methane yield, whether there's an inhibition of methanogens or not. But this was really difficult since the approach was too small, the gas amount was too small, and we had too little repetitions in order to make a significant statement. But then with the help of the qPCR, we could get a quite good idea about this. So it seemed that the methanogens were inhibited quite a lot. Well, are there any questions about this so far? But it's more or less clear? Okay. Well, then we will move to the next part. And this is the last paper that I'm presenting in the frame of this lecture. We will finish it today. And then we start another one where we go in more details about important microbial features that we could use for manipulation of anaerobic microbiomes. This was also one article that I published in the frame of my postdoc thesis. It's called Chemically Stressed Bacterial Communities in Anaerobic Digesters Exhibit Resilience and Ecological Flexibility. Yeah, it's a long title, but we will go into it now step by step. So where we are now with our stressors. So we talked now already about temperature and ammonium and salt. And now we have another set of inhibitors. Now we'll talk about pH, antibiotics, and phosphate. I guess there are, you could make an endless list of possible inhibitors that could be interesting here, but I just used the ones that I was working with so far, and at some point we also have to restrict it to a reasonable amount of information in order to get through the presentation. So what did we do here? We had such a continuous steered tank reactor, which you know already. So we have a vessel, we have a stirrer inside. There are actually 2 stirrers. Here's like a pedal in the middle, and then a bottom stirrer to have it steered very well. Here's a motor unit. Here's an inflow of material. Here's an outflow of our gas that we are producing. The whole thing is double-walled, so we had some hot water inside to heat up our process to mesophilic temperatures. And here was an outlet where we could release all our digestate once we were done, or to fermentate. So we had an experiment lasting for several weeks. And in the first 6 weeks, we tried to kind of normalize the whole system, which was not working too well, but you will see. And then beginning from week 7, we were adding stressors to really disorder the whole microbiome in purpose. And we used in each reactor a different stressor. So we had one reactor with a control. We had another reactor where we were applying nalidixic acid. That's an antibiotic. Then we used GABA aminobutyric acid. It's an organic acid. And we used sodium phosphate. And you see that in each week we were increasing, no, not in each week, but step by step we were increasing the amount of the stressors that we were applying. For example, here we were just starting with 10 milligrams per liter, then we went higher to 0.1 gram, 1 gram, 10, and then again 10. So we tried to increase it continuously in each of these reactors. So that's the result. I suppose you can't recognize anything. It's quite small, but we will move step by step through this diagram and we will enlarge the parts that you're supposed to watch. So let's start with the cumulative methane volume that has been produced. Here you see the weeks. Then with stars we indicated the level of stressors that were applied. And here you already see one problem. We couldn't solve it. So apparently there was a difference in gas production already from the beginning. This happens. So there's always a problem with these reactors. Sometimes they're just not tight, or you're working with really heterogeneous material. So these are just the disturbances you have to work with. So step by step we were actually increasing the organic loading rate to bring it to a reasonable amount of productivity, methane production. And then we started to add the stressors. And then there are some time points, we marked them with red lines. Here's also one at the beginning where we performed the 16S sequencing. So there's not too much to see in this diagram. Apparently the bottom line was working not too good. This was the sodium phosphate. Maybe this was an inhibition, but then you saw that this was already starting before the inhibition, so that's nothing we could argue with. And then we were also surprised that the nalidixic acid and GABA was showing so little gas production in compared to the control, because theoretically both of them are a substrate which could be converted into methane. Well, let's go further through the diagram. Here you see again the methane production, but not cumulative anymore. Now we have an amount of gas that we collected every sample point. So therefore it goes a bit up and down, it's a bit irregular. Here's the start of feeding, so you see that the gas production suddenly increases. Then this looks so chaotic, it's really difficult to see anything here. But there's one thing that you might recognize, which is this line. This is from the GABA. So apparently at week 10, we stressed the microbiome already too much, and then the gas production went down. So in this case, we have been really successful with the inhibition. And here we see the methane ratio. This was also interesting. You see again in the line that represents GABA that it becomes highly irregular before actually the gas production went down. So here you see how it's going down in the gas production per sampling. And here you see that in the methane ratio it was quite irregular. It was very interesting that also the sodium phosphate inhibited microbiomes were quite irregular, because we couldn't make any assumption based on this figure here, because the gas productivity was for some reason so low. But then in the methane ratio, there was really an irregularity which indicates that also this process was somehow disturbed. So, to sum this up, there was far too little gas produced for GABA and nalidixic acid. So, we have indications that there's a process inhibition, either because of the too little gas that was produced or based on these irregularities for GABA. And we could also assume an instability for the phosphate inhibited processes. So what is this for actually? So in the end, it has no practical applicability. We just wanted to compare how a microbiome behaves under different disturbances, under different stressors that we are applying on this microbiome. And we also analyzed this then on the chemical level. Here you see the chemical oxygen demand. And here we distinguish between the total chemical oxygen demand, which means everything in our solution. And here we see the solubilized COD. This means only the COD which was kind of degraded and transferred into a soluble fraction. And here was only one large increase, which was from the GABA. So this indicates very well that there was a process inhibition, because acids were faster produced than they actually could be degraded. But then here's something strange. If we take a look at the total COD, I don't know whether you realize it, but there's something really odd going on here. Think about it for a moment. So I will repeat the question again. These two diagrams do not really fit well with each other. And I want to know why. I want to know whether you see the problem here. So that's exactly the kind of interpretations that you have to do if you're writing one day your own articles. Then you get figures like this, and then you have to make something out of it. You have something? No? So we see that the total COD goes up. Here, the solubilized COD goes also up. So in the case of GABA, the curves fit relatively good together. But then here you see that the line for the antibiotics also go up quite a lot, but they are not increasing in the dissolved COD. So how could this be? What's the reason for this?
Maybe because the one is for bacteria and another for archaea? Archaea?
No. Any other suggestions? You have something?
Yes, maybe I have an idea. I think that the organisms that are creating the soluble COD are inhibited too with the antibiotics.
I have to think about it. this could actually be an explanation, yes? It's a good idea. Other ideas? So I will resolve it. We found references about antibiotics which are degraded not so well, so they can basically precipitate and they can't be accessed by the microbiota. Maybe this does not count for all antibiotics, but it was the case here in the nalidixic acid. So you're adding more and more of the nalidixic acid, therefore the total COD increases. But then the involved microbiota, they are not able to eat it. So it will just remain in the solid fraction and gets not converted into the liquid fraction. So, more parameters. So here we have now the pH. And you see that it goes not too much down in the case of nalidixic acid. So if there was an inhibition, it was at least not through the pH. But then in the case of the nalidixic acid, the pH really went down, which now indicates that we had a high productivity of volatile fatty acids, which was causing before the increasing COD, the solubilized COD. And in accordance to this, we also see that the acids, the total volatile fatty acids, are quite increasing for the GABA-feeded reactors. Okay, now we have a relatively good idea about what was happening in these reactors, how they behaved in regard to these stressors that we applied. But now the main objective was to alter the microbial communication based on the same microbiome or outgoing from the same microbiome and study this. So here you see the whole taxonomic profile. There is not too much to see, or at least it's relatively hard. This is on phylum level. I was surprised by this because I would have assumed that in the case of nalidixic acid, the whole microbiome becomes disrupted. I mean, antibiotics are killing bacteria. But then there was maybe this slight change here in the blue ones and in the orange ones. But apart from this, it looked relatively unchanged. So this was quite surprising for me. So we went to the genus level to make it a bit more clear, to see more specific groups, how they behave. And here you could already see some changes. For example, the samples were relatively different from the start condition. Then most of them had a lot of this Trichococcus. You see that the sodium phosphate was really going out of line. So apparently this was really affected on the taxonomic level. Well, and then compared to the control, nalidixic acid and GABA was little affected, which was also surprising to me because they were actually quite inhibited, at least in the case of the nalidixic acid. So this was really difficult to see actually. Well, we now performed a principal component analysis. Maybe you remember that we talked about this. I can't explain you the mathematical details, but this will also take quite some time. But maybe I can simplify it a bit, that you imagine that you are calculating for all these shifts here, for all these data points, like a cloud of variance. And then you're moving this variance in a three-dimensional coordinate system in order to find a perspective where you see the biggest difference between these data points. So we did this. So we included all these taxonomic data into the algorithm to perform this principal component analysis. And then we got this. how would you make an interpretation out of this? What do you see here? Think about it for a moment. You're not in a rush. I mean, there's nothing which would really surprise me, but if you just try to explain descriptively what you're seeing here. Well, we see that the inoculum is really different from all the other samples. So this shows that there was some kind of evolution taking place in our microbiome, and that we really changed it based on all the procedure that we had here. We have several days. This is actually day 0. And then we have day 56, 70, 77. And here one cloud which behaves more or less the same, but then the sodium phosphate really gets out of range from the other one. So it seems that especially the phosphate had a huge impact on the microbiome, on the composition of the microbiome. This was quite interesting to see. Another possibility to analyze such microbiomes is to apply Venn diagrams. I suppose that you have seen such diagrams before. The Venn diagrams are quite common, not just for analyzing of microbiomes. Well, and you see here these kind of circles. And this was at day 65, so relatively in the beginning of our inhibition. And then you see numbers. And these numbers indicate which organisms were changed significantly. There were surprisingly little changes in the GABA. But remember that this was still at the beginning. The GABA was not too suppressed at this time yet. So there were quite some changes at the nalidixic acid. This was interesting because this was not so clear in the principal component analysis or the taxonomic profile. So apparently there was also an effect on the nalidixic acid, also a bit difficult to realize. And then we have the phosphate. The phosphate was inhibited quite a lot compared to the other ones. You have green numbers and red numbers. Green means some organisms are increased in their abundance, and some organisms are actually decreased in their abundance. And here you have, for example, shared microorganisms. There are 2 microorganisms that were changed significantly as well in nalidixic acid and as well in phosphate. So this is a good strategy to find indicator microorganisms that could be used to maybe one day indicate process disturbances in such a reactor. Well, now we have the other days, day 70, day 77. Well, and you see that the number increases more and more, especially in phosphate. In the end, we have 66 organisms that change their abundancy significantly based on this manipulation. Okay, so now we also described the microbiome involved, and now comes the really interesting part, because now we performed a Lotka-Volterra modeling. I mentioned this before, and this was this model with the wolf or the fox and the rabbit. So if the rabbit grows, the wolf has more to eat, then the wolves will grow. If we have more wolves, then they will eat more rabbits, and the rabbits will decrease. And like this, there's always an interdependency between the wolves and the rabbits. And you can express this mathematically. So here you see the growth rate of one of these species, for example, the wolf or the rabbit. And this is now equivalent or equal to a certain abundancy of wolf or rabbit. in regard to an intrinsic growth rate and an interaction coefficient. Hi, please come in. Multiplied with the abundancy of the other species. So like this, you have a dependency of the abundance of one species from the other, especially based on this interaction coefficient. So this is quite interesting, and with this alpha, with this interaction coefficient, you can actually say whether they grow with a beneficial effect, whether they support each other, whether they are competitive, or whether they don't care, whether they just exist next to each other and nothing happens. So now we have applied our taxonomic profiles on this formula, because you can add actually several of these growth rates, and you can create larger independencies for several organisms. This becomes relatively complicated. For this, we had again a bioinformatician. And then we produce this figure. Oh, here are still the German words. Gray negative, green positive, yellow no interaction. The colors are actually not too easy to distinguish. So you have yellow colors, you have green colors, and this is supposed to be gray. So what do we see here? We have different organisms. That's the one that we were checking. Those were relatively abundant organisms. And then we were applying this Lotka-Volterra model. And then you can, for example, see that Recanalaceae is actually suppressing, or at least not beneficial for the Trichococcus in the first condition, which is the control. But then suddenly, when we added our stressors, the nalidixic acid, the sodium phosphate, or the GABA, suddenly they started to collaborate. So this was quite strange. And this is not just one case. You can see this for all kinds of combinations of microorganisms. So in one condition, they seem to collaborate, and in another condition, they seem to fight against each other. So what does this mean for microbiome manipulation? It means it's extremely difficult to find a very well-defined procedure to change a microbiome in purpose, because whatever condition you can have different relation between the involved microbiota. And this could mean that the whole microbiome behaves differently. Is it more or less clear? Yes? Okay, great. Well, so with this I would like to summarize this, not just this article, but the whole chapter. So we have seen that there is a separate acidification which can be robust against heat shocks up to 70 degrees in order to minimize metagenic contaminations. We have seen that high nitrogen concentrations can stabilize the pH, which is a problem in dark fermentation, because then we don't reach a low pH which could inhibit methanogens. We have seen that we can apply a combination of nitrogen removal strategies in dark fermentation in order to better control acidification, which allowed us in the end to increase the acid yield. And we have also seen that even though the pH is not going down in order to inhibit the methanogens, that we can still inhibit the methanogens when we apply a very high concentration of nitrogen. It might be interesting now to combine actually these temperature shocks with our nitrogen-rich input concentration in order to inhibit methanogens further. That's what we haven't tried yet. And we were really surprised to see that in the chicken manure-based dark fermentation, we reached very low concentration or very low cell counts for bacteria, which were still in the same level of efficiency like the control without any inhibiting effects for the bacteria. Well, and then in the last article now we discussed several stressors and have seen how they affect the underlying microbiome, that we have quite dynamic interaction patterns which make it really, really difficult to propose any targeted strategy, any clear targeted strategy to actually manipulate anaerobic microbiomes. For this we have used this Lotka-Volterra model, and you also have seen some methods that you can use to compare microbiomes, like the Venn diagram, or this principal component analysis. Oh, this is wrong. This should not be skin. This should be principal component analysis. I have to correct this. Okay, that's all. Are there any questions? Meanwhile, we can already change the presentation. Can you please add a new presentation? Until we have it, you can think about whether you have any questions. Okay. So I'm actually jumping over one of the chapters. Theoretically, we would now start with the proteomic profiles. We have already seen one, so maybe it's not too important. Here's actually a second one, and I'm also going a bit into the methodology. But I found it really important that we managed to finish chapter 7, because in the end of chapter 7, we are coming back to the photosynthesis. And after we spend so much time explaining how photosynthesis works, we should now also see what's actually the relation to anaerobic microbiomes. Well, so we will do this, and then if I suppose we need time until tomorrow to finish this. And if we have some time left in the end, we can still talk about the proteomic profiles. So let's get started. I will give you a short introduction into the microcosm, into the term microcosm. And then we will see some interesting microorganisms for anaerobic digestion. So far, we were not really going into specific microorganisms. We always remained on a taxonomic overview of several groups. And now we will be a bit more specific. We will ask the question how to manipulate anaerobic microbiomes before we then come to anoxic photosynthesis, light-enhanced anaerobic digestion, and the impact of light on anaerobic digester microbiomes. So let's get started. This is actually a modified version of my postdoc defense. I changed something, so it's not the same, but yeah, it's similar. And we will start with exotic high-performance organisms for anaerobic degradation processes. And the goals of this talk are basically to motivate environmental engineers for anaerobic digestion and anaerobic microbiomes, to increase your understanding of the functional diversity we have to deal with, because this also affects us on a technical level in the end, and to highlight exotic microbiomes and features which might become interesting in the future. Not all of them, some are quite basic, more related to basic research, but some of them are already quite interesting to improve anaerobic digestion processes. We don't know, we just don't know yet how. Well, the biochemical taste you got already. So that's biogas plant. That's what you know already. So I think here I'm not explaining you so much new. It's the simplest scheme that you can have of a biogas plant, a vessel which can receive waste, produces biogas, and in the end we have our digestate. So in these last days we were talking a lot about the underlying microbiomes, and although we got some idea what's happening there, maybe you would still agree to me that it's a black box. We have a rough idea, but somehow we are still dwelling into it, and it's not all clear yet. So we are just at the beginning, scratching at the surface, and I think there remains a lot of work to fully understand how to work with anaerobic microbiomes, how to manipulate, how to change them. Well, in this regard, I would like to show these pictures, which I liked very much, and I saw them once when I was on Twitter. Now it's called X. But then I saw these pictures, and those are actually petri dishes. And they were inoculated with microbes in such a way that they look like art. So here you have two which are representing a cell-to-cell contact. Here you have bacteria which grow in the form of neuronal cells. And here you even have fluorescent microbes that were used to draw something which is supposed to look like the San Diego beach. I don't know whether you can recognize the San Diego beach there. So when I saw these pictures, I became really interested in this, and I searched for more. And then I found this really nice webpage called Earthly Mission. I hope it still exists. I checked it last time, 2022. And what you're seeing here are petri dishes, which are inoculated in such a way that they look like planets. They were changing the background for stars, then it looks more star-like, but yeah, then the petri dishes are supposed to represent planets. It even gets bigger. This is a picture from the NASA, and they wanted to draw something which looks similar to the small Magellanic Cloud, which you see to the left, and here to the right, their interpretation based on microbial art. So I really like these pictures, and at this point I was wondering why people are so eager to draw cosmic structures with microorganisms. And of course the answer lays in the term microverse, because the deeper we go into the microbiomes, the more we are magnifying things, the better our technologies are, the more complex the whole thing is. And then it reaches a complexity which is almost on a galactical level. That's why we use this term microverse. Here's a nice comparison. We already had this comparison to the population in Germany, that we have many more microbes in 1 milliliter of sludge than inhabitants in Germany. Now I brought this to a cosmic level, and I estimated the number of stars. No, this one I got from Google, but I estimated the number of microorganisms in a digester. And I was surprised that we have many more microorganisms in a digester than stars in our entire Milky Way, in our entire galaxy. So this again raises the question, how can we mobilize this huge workforce in order to work fast, do something useful with it, maybe something else than just biogas, for example, producing organic acids and hydrogen. Well, this idea, this research started actually with the invention of high-throughput sequencing. And this is relatively new. So when I studied, this was until 2013, it's not too long ago, high-throughput sequencing was not so well known, it was just emerging. Now we have these nanopore sequences, which work quite fine. Well, and with this now we have like a playground for microbiome research, which really is interesting just on a research level, but hopefully we can also do something practically out of it. Well, and doing this research, we found now already many interesting microbes. People sometimes say we opened a treasure box full of microbes, and now we know they are there, we just don't know how to exactly use them. And here I made a list for you. This is a list of special microbial features that we're now going to discuss step by step. And there's one which you know really well, the ATP synthase. What was the ATP synthase? talked about it in regard to photosynthesis, but also glycolysis, citric acid cycle. What does it do? No one has an idea? Talked already like 4 times about it. I will just ask someone. What would you say? What is the ATP synthase? You need this is not working, your microphone?
Well, I think it's an enzyme. My memory isn't good. Okay,
it's an enzyme, that's right. What does it do?
Well, synthesize
Yes.
the ATP.
That's right. And the ATP was what again?
Adenine triphosphate.
Okay, great. That's absolutely right. So let's go through it step by step, and we start with the ATP synthase. Here's a nice video. It's a bit dark, but they try here to visualize how the ATP synthase actually works. So you see how it's turning, how the protons are going through. And this movement is then actually used in order to press the ATP and the phosphate together in this head unit. Here at the bottom you almost see how the ATP is produced. Well, what's so special about it? I made a very small video to better highlight what the ATP synthase is doing. And you can really imagine it like a power plant, like a turbine. And protons are running through it in order to power it. And like this, we are getting the energy that our cells are going to use. There are always 3 protons. Across life, you always find 3 protons which are used to produce ATP. But then suddenly in methanogenesis, I think it's about the syntrophic bacteria actually, it's not enough anymore. So the gradient in the protons is so little, or they can just build up such a little gradient in protons with the conditions that they have there, that it's just not possible to produce the ATP there. And they evolved with a very rare case of ATP synthase, which is now using 5 protons instead of 3. And this brings it very close to the energetic minimum where life can still exist. And this is a really interesting case for basic research in biochemistry. And there is not too much published about it, but there is an interesting article which is related to Star Wars, which is called The Past and Present of the Sodium Energetics. May the sodium motive force be with you. So this is now another interesting thing. So we not only have a ATP synthase, which is able to use a very different amount of protons, which is quite unusual and which allows to work at the minimum of life, but we also have suddenly an ATP synthase which can work with sodium instead of protons. So this is an extremely rare case. So biochemically, this is really interesting to investigate and makes it once again a really interesting playground for microbiome research. Well, now we are becoming a bit more applied. And here we're seeing a taxonomic profile. But this taxonomic profile was actually done with proteins, not with DNA sequences. So we would actually explain this in detail in the proteomic chapter. Maybe we can talk about it tomorrow in more detail. But you see here a lot of different microbes. And then there's something interesting. It's the bluish one here. What are actually bacteriophages? Do you know this term, bacteriophages? Have you ever heard about it? No? Do you have an idea? No? Okay, so, but you all know what a virus is. So basically a bacteriophage is the same as a virus, but it attacks bacteria. So bacteria also can get sick. One day I was asked whether an anaerobic digester could get the flu. Well, I don't think so, but certainly the bacteriophages will have a huge impact on the underlying microbiome. But they're really hard to detect. So here you see a very thin bluish line next to all the other microorganisms. Why is it so thin? Well, if you compare the size of a virus or a bacteriophage to a bacterium, it's extremely small. So this means that you have very few proteins related to the bacteriophages. But then if you compare the total number of bacteriophages to bacteria and archaea, there could be a lot of them. So in this work from 2019 from Haya et al., they were speculating on how this actually affects the microbiome. And this has not been proven, it's just an hypothesis, but I think it sounds somehow logically what they're proposing here. So there are auxotrophic microorganisms and prototrophic microorganisms. The auxotrophic microorganisms, they don't grow very well. They have very special requirements, and usually they would just not grow. The prototrophs, they grow very easy. They need a carbon source and, well, not much else, and then they will just grow. The bacteriophages, they are now attacking both likewise. And when they are attacking the prototrophs, then they are destroying the prototrophic cells, and all the input material is released into the environment. So suddenly you have a lot of specific nutrients which become available for auxotrophic microorganisms. So maybe the bacteriophages are the basis that auxotrophic microorganisms can grow at all. We don't know, it's just an hypothesis. But it's one possible strategy to explain what might be the impact of bacteriophages on anaerobic microbiomes. And I would say it's an exotic example because bacteriophages in regard to anaerobic digestion are very hardly addressed. So there are very few articles which are talking about bacteriophages, but I think it would be actually a quite interesting concept to manipulate anaerobic digesters. So it would be interesting to just culture some bacteriophages and add them to the process in order to cause some process disturbances and see how it affects the microbiome in the end. Well, the next feature that I want to talk about is the so-called CPR group, which stands for candidate phyla radiation. Well, this refers to microorganisms which are mostly uncultivated. So they are very hard to culture, very difficult, therefore people don't like to work with them. People like to work with microbes which are easy to culture. Sometimes they're also known nanobacteria or ultra small bacteria because they have very small genomes. But there are many of them. So originally they were representing 70 different phyla, and they are actually representing 15% of all microbial diversity. I mean, probably you can't generalize this. This probably also depends on the area where you're working. But apparently there are many of them. And since 2018, this has changed, and now all these 70 phyla are combined to one group, which is now the CPR group. Why is it important? I mean, they don't look so special. They have small genomes. They lack a lot of several pathways. They are therefore not able to survive on their own. Why should we be so interested in them? Well, there was a very interesting article discussing contamination with petroleum in the Gulf of Mexico. And they were analyzing this taxonomically, and then they found this Gracilobacterium. They were describing the biochemical pathway. Don't worry, you don't have to read any of this. The only thing that I want to show here are these red circles. And they highlight important pathways which are inhibited. So it's really a disabled microorganism. How does it survive? So there is a host organism called Colvellia. And for some reason, this Colvellia keeps this nanobacterium alive. Why is it doing this? We don't know. I mean, it could be that it has some kind of hidden superpower, for example, to make the petroleum better accessible and degradable. And from this also the Colvellia would take profit. So it would make sense to keep the nanobacterium alive in order to access new resources that could be degraded to grow. So I'm just speculating here. We don't know yet, but it's definitely interesting. And so far, there are no articles. At least I haven't found any articles about nanobacteria and anaerobic digestion. I was looking already for it. It's really interesting. There was one article that I found, and they were comparing microorganisms in an alkaline spring, so not in a biogas plant, but it was an anaerobic environment where they find in all the chemolithotrophs some methanogens. And then in all the other prokaryotes, they also found this Gracilobacterium. So apparently, nanobacteria can coexist with methanogenic archaea in anaerobic microbiomes. And I'm just wondering whether this would also take place in a biogas plant, how it would affect the biogas plant, and whether we could use this someday to maybe turn the wheel of the efficiency a bit further in order to make our biogas plants more efficient. Okay, we have 45 minutes left. Well, here's a complicated name, Neocalammasticomycota, but you can simplify it to anaerobic fungi. So they exist, and we don't know this for so long time. So they have been, I don't know, a few decades ago, they have been found. And I was surprised by this because you could actually recognize them under the light microscope. They have a growth form which is quite specific, but still it took a long time until we found them. And they are actually important in many animals. So you find them in many animals like horses, cows, and so on. And now there are a few studies which highlight that they can be found in anaerobic digesters, but apparently not in all of them. And a very interesting capability of them is to absorb hydrogen. What are they doing with the hydrogen? Certainly they will not consume it. They can't consume it. They are not methanogens. So if you want to make something with the hydrogen under anaerobic conditions, it should optimally be methane. So how is this important? Maybe you remember that I told you that there is an inhibition of hydrogen on syntrophic bacteria. So if the hydrogen level is too high, the degradation of longer fatty acids into smaller fatty acids and hydrogen can be inhibited. That's something that we would like to prevent. And maybe they are helping here. Maybe they are colocalizing with our syntrophic bacteria and with our methanogenic archaea. And then upon release of hydrogen during the syntrophic degradation of organic acids, they might be able to take up this hydrogen, and then they could feed it on demand on the methanogenic archaea. It's a hypothesis. Again, we don't know. Another really interesting feature about them is the so-called, cellulosome. You might have heard about it. It's actually quite common in some bacteria. But they have a really efficient cellulosome, and they have a really efficient way to use it. So this picture is quite overwhelming. You don't have to understand all these units here. They are named, but it's not so important. What's important is that you have many of these units. So it's like an assembly of many enzymes that are working together in a concerted degradation reaction to access substrates which are difficult to degrade. For example, our cellulose or our hemicellulose. Well, as I told you, some bacteria have it as well, but they have a really interesting mechanism. So first of all, they're able to release the cellulosome into the environment, but they can also keep the cellulosome bound to the cell surface. And if they keep it bound to the cell surface, they can grow into the substrate. You know that the fungi that are growing with these kind of hyphen, and they can actually penetrate into the substrate. And once they do this, they can degrade the material with their cellulosomes. So they could be highly efficient in the access of biomass, which is difficult to degrade. And at the same time, they could help us to prevent inhibition for the syntrophic bacteria in order to accelerate the methanogenesis. So I think this is highly interesting, and we now know that this is possible, but we still don't know really how to enrich them. So this is still going to be research for the future. So here we have another case, the so-called endosymbiotic theory. Who has heard from the endosymbiotic theory? Who knows it? You know it?
It's a theory for in a moment in the time, ancient bacteria or microorganisms consume another more small, and their synergy of both creates a new mechanisms of for survivor.
Yeah, maybe we could say it in that way. So can you repeat it? I don't get it fully what you said.
Yes, that in a moment in time, a microorganism,
Somewhere in the past.
and maybe a few a time for Luca. I consume that microorganisms
Yes.
consume another more small,
Yeah.
and both grow up, and that's the endosymbiosis. Grow up together.
It's more or less right, yeah. So, I mean, it doesn't need to be a microorganism. Well, yeah, it should, but it could be, yeah, you're right. It could be, it's microorganism, but it could be a eukaryotic cell and also a prokaryotic cell. In both, we have endosymbionts. And yeah, for example, our mitochondria, the organelles which we use to breathe, our cells are breathing with this, are actually in bacterium, which somewhere in time was swallowed by a eukaryotic cell but not degraded. And the same goes for the chloroplast, which we know already from the photosynthesis. Well, how is this now related to anaerobic digestion? There's a really interesting case. There's an organism called Metopus paliformis. It's an amoeba. It's a eukaryote. And I told you that we find almost no eukaryotes in anaerobic digesters. But there are a few, usually less than 1%. And amongst this 1%, we have the Metopus paliformis. And it's actually full of methanogenic archaea. There are two pictures, and one highlights all the methanogenic archaea inside. And this other one highlights something really strange, so-called hydrogenosomes inside a cell. So it seems that it's likewise to the anaerobic fungi able to absorb the hydrogen, store it in these hydrogenosomes, and then we have all the methanogens surrounding this, and then probably they are fed by this eukaryote. There's some kind of synergy, so they can also take profit from the methanogens. I don't know yet what is the reason, but there is a relation. But I think this is amazing, because imagine that you have an anaerobic digester, and the anaerobic digester gets too acidic, and maybe most of your methanogens die. But then maybe there are some which are contained in our Metopus paliformis, which keeps them alive and happy and well fed. And then after the acid shock is surpassed, then they could regrow from this Metopus paliformis. So just one idea. And also it could reduce the inhibition of syntrophic bacteria once again, which are easily inhibited by the hydrogen. So this is actually not too new, but sadly there is very little published about this. So that's why I also would say it's an exotic case in the anaerobic digestion research. And there's one article, for example, from 2008, anaerobic protozoa and their growth in biomethanation systems. And they found that with a higher abundance of these eukaryotes, the process becomes more efficient, the degradation rate increases, and they also described several of them, which seem to be important to have a stable system. What's also interesting that they were able to make some lab experiments where they depleted some of the methanogens inside the protozoa, and for some of them the growth rate was depleted up to 70%. Not all were affected, but some of them are highly dependent of the methanogens inside, so quite interesting. Well, the next phenomenon you know already very well, which is the direct interspecies electron transfer. You also have seen these pictures. And this is actually one of the first pictures where direct interspecies electron transfer has been shown. This is from 2006. So I told you it's a quite young research field, and we are just getting started with this. This is one of the electron graphs that I did. And here, maybe remember that I supposed that this could be methanogens. And here you see some bacteria with fibers. Maybe they are just connecting to the methanosacina using the direct interspecies electron transfer. We don't know. So it's interesting that the direct interspecies electron transfer is actually not just important for anaerobic digesters. You could come into contact with it also in other fields of research. For example, in composting. There are very few articles which indicate that you could put electrodes into a compost in order to change its activity and maybe also accelerating its decomposition rate. Then we have microbial electrolysis cells. Maybe you remember this picture that I have shown you from an anode and a cathode that we put into an anaerobic digester in order to foster this direct interspecies electron transfer and to accelerate methanogenesis. You could do the same here, but if you're able to have here a selective membrane, a semi-permeable membrane, which does let pass hydrogen protons, but no bacteria, then you would have here a chamber with protons, and with the right catalysator on your cathode, you would be able to convert the hydrogen, the protons into hydrogen. So this is actually a relatively nice concept to produce hydrogen. And this is also relatively new. People doing research on this since 2005. So it's also a relatively new concept. And the last one about BTEX plumes. So these BTEX substances are quite problematic because they contain a lot of aromatic compounds which are usually really difficult to degrade under anaerobic conditions. But it was found that this direct interspecies electron transfer actually facilitates the degradation of these substances. And you could put some conductive particles together with some oxidative substances into your BTX plumes. And like this, you could foster the direct interspecies electron transfer in the soil that you want to renature, and then you would see a faster degradation of these contaminants. Yeah, here's the reference for this picture as well. Well, then we're talking about nanostructures. These are also highly interesting and not too much published yet. Therefore, again, I would say it's a quite exotic case. And well, we have seen already this. So this was a kind of conductive crystal fibers which interact with the methanogens, the green dots here, in order to perform this direct interspecies electron transfer. Well, but there are other effects that we can cause based on nanostructures. And there was a really interesting article from 2017 where they used so-called carbon nanotubes. So they are relatively expensive, so maybe it's not really interesting for the industry. But what they have shown is that the carbon nanotubes are increasing the efficiency of methanogens involved. And then they were looking for specific signs of this direct interspecies electron transfer because it's also kind of conductive, and they were expecting that diode will take place here, but they didn't find it. Moreover, they saw an improvement for methanoculeus in pure culture. And maybe you remember this methanoculeus, this hydrogenotrophic methanogen, for which so far no diode was shown. So apparently here's an improvement which we can't explain, at least not by the direct interspecies electron transfer. And I'm eager awaiting that anyone resolves this mechanism. This goes quite deep into biochemistry, but it's really, really interesting what's happening there. Well, then we have the phototrophy. I think I have shown you this picture already in the very first presentation where we talked about photosynthesis. And this was just a lucky incident in summer. And we will go into this in the further part, in the second half of this talk, in detail to see what can be found in such a phototrophic biofilm. Then communications are maybe not that exotic, but I mean, as this hasn't been highlighted so often, these dynamic communication patterns where microorganisms can cooperate with certain microorganisms, and in another condition they fight against each other. This dynamic behavior was not highlighted so much. Therefore, I also regard it as an exotic case, but maybe it's rather common. Maybe we find it everywhere where we have a lot of microbes involved. And also the communication in general is extraordinary complex, especially if we take into account, I mean, you remember this map that I've shown you before on quorum sensing. Without going into detail, what we are seeing here, we just see many pathways for many different molecules that can be transported via the cell wall in order to tell other microbes how many other microbes are there, so they can sense each other. And this is just the tip of the iceberg. Now imagine that we have thousands of microorganisms involved, which is the case in anaerobic digestion, which again makes anaerobic digestion a really interesting playground for microbiome research to understand the complexity of microbiomes. Well, in the last case, you know already, we already mentioned it when we talked about the capabilities of microbiomes, the capability of memory, that they actually can safe somewhere on a structural level, that there was somewhere a certain selection pressure they had to deal with. And then the next time when the selection pressure comes up, they can react much faster. Yeah, this picture you might remember as well. So we have seen that in a biofilm, a small light beam was used to irrigate a small region, and then they opened their potassium channels, which remained open after the light beam disappeared, and now they can actually react much faster when the light beam reappears. Okay, with this I would like to summarize this. So we have seen some unusual enzymes in regard to the ATP synthase, which are given in anaerobic microbiomes, which is quite interesting for the biochemical research. We have seen some quite unusual microorganisms, and they could play a major role in order to improve anaerobic microbiomes, namely the CPR group, anaerobic fungi, and the symbionts. We also have seen that light might have positive impacts on anaerobic digesters. Or well, we don't have seen it yet, we just have seen this phototrophic biofilm, but we will see that it might have a positive impact. So this could be a new selection pressure, but also the application of electricity, nanostructures, or even bacteriophages could be a really interesting selection pressure to change and manipulate anaerobic microbiomes in the future. Well, we also have seen a really high complexity behind the microbiomes in terms of communication, how they socialize, they even live within each other. And advancing research just reveals more and more complexity that we have to deal with. Which brings us again to the term the microverse. And really important, anaerobic microbiomes do not only contain bacteria and archaea. And usually this seems to be the case because they are most abundantly, of course, more than 90% are archaea and bacteria together. But then you have a few percent of the other organisms, and they seem to have a really important role in anaerobic microbiomes. We just have to discover it and do more research to understand what's going on here. Yeah, but how to manipulate microbiomes, now we don't really know. I told you we have so many microorganisms involved. We have 300 key players, but then thousands of, let's say, side players which are involved. And we don't know how to control this complexity really. And I made this sketch to better highlight this. So you all know the wolf and the Red Riding Hood. And then the Red Riding Hood represents a Clostridium, a new bacterium that we might add to the process. And it says, well, I'm a new Clostridium and I want to help to digest the substrate better. And then the wolf represents the organisms that are already there in our microbiome. And it says, yeah, come closer, I need to see you. And then it will just consume the Clostridium, which means that our bioaugmentation is likely to be unsuccessful. So this again highlights how difficult it is to manipulate anaerobic microbiomes. Well, now we come to another topic, which is in regard to light. We have 25 minutes left, so maybe we can come through the introduction. Oh, so were there any questions before? Everything clear? You could follow it? Great. So that's the abstract. You can read it later on if you want. I will explain all what is necessary, so it's just for the completeness of the slides. That's the title, Shedding Light on Biogas. Phototrophic biofilms and anaerobic digesters hold potential for improved biogas production. So what did we do here? Let's start with the question, what is photosynthesis? photosynthesis in bacteria. Does this happen? And shedding light on biogas, does this make sense? I think we can go relatively fast through the photosynthesis because you know it already. Just to remind you, we have the light-driven reaction, we have the Calvin cycle. Then we have this electron transport chain in order to convert the light into, or to use the light for electrolysis of water. And with this we produce protons and oxygen. And then we use the protons and the electrons to create a gradient of protons, which can then power our ATP synthase. And once the electrons lose their energy, we can recharge them in the photosystem 1 in order to transfer them together with protons on the NADP plus in order to, or which is an important electron carrier to build up biomass in the end. So I hope you still remember this. We have one problem because there are no growing plants in anaerobic digesters, at least not the green ones. So all this concept is also known as oxygenic photosynthesis. Could this play a role in anaerobic digestion? We don't really know, but the only organism that comes into play here are cyanobacteria. Sometimes we find them, and I don't know whether we could use them for photosynthesis in anaerobic microbiomes. There is no work that I know about it. So they could actually perform this oxygenic photosynthesis. Well, but apart from this, we have anoxic photosynthesis. And there are some major groups, which are the purple bacteria and the green bacteria. You may have seen them already. They can be further divided into Rhodospirillaceae and Chromiataceae for the purple bacteria, or into Chlorobiaceae and Chloroflexaceae for the green bacteria. How are they different? First of all, they have different structures to perform the light reaction. In the case of the purple bacteria, we talk about involutions. So basically, all the components for photosynthesis are implemented into the membrane. And then we have so-called chlorosomes. Those are vesicular structures that contain all the proteins, and they are basically attaching to the membrane of these green bacteria. They are different in the number of pigments which they have. So the purple bacteria basically have bacterial chlorophyll A, few species with bacterial chlorophyll B, and roughly 350 different carotenoids. Well, then we have other bacterial chlorophylls, C, E, sometimes D in green bacteria. They also use carotenoids, but then they also use polypeptides as pigments. So it's assumed that they have a wider range of wavelengths which they can cover to collect their energy. Well, and then we have the antenna complex, which you also might remember, the structure where we perform the electrolysis in the end of the water. And this is much more simple in the case of the purple bacteria and more complex in the case of the green bacteria. So I would assume, I don't know for sure, but I would assume that the green bacteria are just more efficient in collecting light. But maybe the purple bacteria are just more dynamic because they don't need so complex structures, and maybe it's easier to build them up once there is light. So they can maybe more dynamically change between phototrophy and chemotrophy. So that's now the electron transport chain. And now you have a really important task. You have to tell me what's the difference compared to the oxic photosynthesis. Have a look at it and let me know what you think. No one has an idea?
Sorry, but can you repeat the question, please?
Yes. This is the anoxic photosynthesis. The light reaction from the anoxic photosynthesis. And what is different here compared to the oxic photosynthesis, the one that we find in plants? Because this is most likely the mechanism which is interesting for our anaerobic microbiomes, unless cyanobacteria could be involved. But apart from this, all the other bacteria will use this concept.
Could be that inner interest of this is that occurs or happen in the cell wall.
I mean, I want to know what's the exact difference between both pathways. So the plants are also doing it in a kind of inner wall, in the chlorosomes, chloroplasts, in the thylakoids. So it's also a kind of intracellular system. But if we just take a look at these different components, what's the major difference here? Yes?
Well, first of all, I haven't read something like this, but I remember when an oxic system, the oxygen is not the ultimate receptor of electron. So in this case, I could see maybe it's about the something. The difference is in hydrogen.
Yes, that's right. I mean, anyway, in photosynthesis, there is no oxygen as final electron acceptor. This would just be in the respiratory chain where we have the oxygen. But here in the photosynthesis, usually we have this electrolysis of water. And you're right, I don't see any electrolysis of water. This means there is no release of protons and no release of electrons. But that's what we would need in order to produce more substrate for methanogens, in order to chemically reduce more carbon dioxide. So that's something that we will probably not achieve. I don't know whether there is another mechanism, but it looks on first sight not too promising. So where do they take the electrons from? So it seems that an electron is always the same, and it's just cycling over and over again. And while it is cycling, it produces a gradient of protons, but it's not producing the protons. So there is no electrolysis. The protons have to be there already, for example, from acids or whatever. So there must be protons present which can be used based on one electron, which is always cycling in theory, in order to activate this pump, this cytochrome BF complex. Here it's called differently, here it's BC1, in order to pump the protons inside and to create this gradient of protons, which can then power the ATP synthase. Well, here you see a picture of the pigments that I have of the photosynthetic apparatus. On the left side, you see the purple bacteria. Here you see the green bacteria. So they look a little bit different, but work really similar. So now the question, why should we be interested in applying light? What would you think? What could we achieve when we are applying light to anaerobic microbiomes? Any guesses? Well, one answer is already there. Could photosynthesis reduce CO2, chemically reduce CO2? Like this, we could create more methane, we would have less CO2, and we would have a higher gas quality. This could be interesting. We don't know whether it works, because this comes to the question whether light could actually help to produce additional biomass. For this, it must be able to perform electrolysis in order to have hydrogen which could reduce the CO2. At least in the anoxic photosynthesis, this is not the case. So it's unlikely that this will happen. But maybe there is another mechanism that we still don't know. Maybe they could improve the degradation. Maybe the light could be used with some strange mechanism to increase the oxidation potential, which might make certain substrates more accessible, like BTEX compounds, like hemicellulose, all the substrates which are difficult to access. We could raise the questions whether microaerophilic conditions could be formed. This could be very interesting because literature shows that microaerophilic conditions can increase the degradation efficiency. But again, for this, we would have to perform an oxygenic photosynthesis. And again, this would only be possible with cyanobacteria, and I don't know whether they are going to do this under anaerobic conditions. Maybe light could help to improve the diet. This would be interesting. So you know that we are always pumping electrons from the syntrophic bacteria to the methanogens. And why not increasing the energetic level of these electrons to improve the transfer, to make everything a bit more faster? This could happen. And it could also affect the methanogenic pathway. That's not obvious, but there are some hints in the literature that this is actually taking place. There's an article, it's actually quite old, from 1991, and it's about the methyl coenzyme M reductase. Maybe you know or you remember that we talked about it. It's like the very final step of methanogenesis where we have the methyl coenzyme M. And chemically it looks like this. We have the methyl group, and then there's the coenzyme M, which is a large chemical structure. And in the last step, the coenzyme M can be transferred on coenzyme B, and by this the methyl group binds another proton or another hydrogen atom and is then released in the form of methane. It has been found that actually this reaction, this release of the methyl group from the coenzyme M can be triggered by light. So this is quite interesting. So it seems that there is a direct impact of light on methanogenic pathways, but this has just been shown for Methanothermobacter, Thermoautotrophicus, and it has only been shown in vitro, but not in vivo. So they don't have shown it with the organisms themselves. They rather extracted the whole compound, then they put some light on it, and then they saw that this was happening. Well, I'm not the first one with the idea to apply light. There was already a patent in 1999. Yeah, for some reason it did not come into the industry. One reason is that it's really difficult to lighten up an anaerobic digester. Usually you have a thick wall of concrete, and then you have this dark sludge inside. So it's really difficult to illuminate this. This might be one reason that it didn't really come into the industry. But people saw the possibility to do it and were patenting it. So here you see a very simple scheme. You have here the waste tank. The waste goes into the digester, which is then lightened up. Then you have your gas collection tank, a separator where you collect or recollect the bacteria to use them later on. And you have the liquid collection tank with your cleaned water. It's just one concept which indicates that it could be possible to apply light here. Well, and this brings us to this picture about photosynthesis in anaerobic microbiomes with this reddish biofilm that luckily showed up in summer doing experiments that we did at the Robert Boyle Institute when I was still working there. Well, and we were scratching this reddish biofilm from the wall. So we were actually removing all the sludge, and then we were scratching the reddish biofilm in order to analyze it. The setup was relatively simple. So we had this reactor. It was basically filled with seawater sludge and untreated grass as a substrate. And it was a two-stage system, actually, built out of plexiglass, so it was transparent. After a few weeks, a biofilm appeared. We performed 16S sequencing and also shotgun metagenomic sequencing. I will not go into the 16S sequencing. If you're interested in this, you can read the whole article, but I think the metagenomic sequencing is by far more interesting. So we will talk in detail about the metagenomic sequences. And we did here a taxonomic analysis, and also with the help of bioinformaticians again, a functional analysis. So maybe the time is not enough to start this now. So I would not like to stop with this somewhere in the middle. So I would suggest that we are done for today, and then we finish this off tomorrow. Are there any questions? No? Okay, then thanks a lot. See you tomorrow. And well, good luck with the preparation for the exam. It's a lot of material. I hope it's somehow feasible for you. Let's see. Bye, see you tomorrow.